Okay, so I think it's recording now. Maybe. Seems to work.
We can probably start.
Yeah.
So, hello everybody. The speaker of today's colloquium is Emmanuel Barillot. Emmanuel is the director of the Computational Oncology Department, which is a joint affiliation with the Institut Curie in CERN and also in PRAIRIE-PSAI. He directs the Computational Systems Biology of Cancer team, which is focused on understanding tumorogenesis and tumor progression using computational approaches and developing methodological approaches when needed. Emmanuel is also co-director of the Institut Curie Bioinformatics Core Facility, and he has published more than 200 papers in different fields, going from bioinformatics to cancer biology to computer science. And today we'll be talking about multimodal modeling of tumors and digital twins, and we're looking forward to your talk, Emmanuel.
Okay, many thanks for the introduction, Laura. So thank you for the invitation. So when preparing this talk, I was hesitating about how to orient it, given that, I mean, in PRAIRIE, there are many people who are not familiar, I guess, with oncology and computational oncology, if not computational biology. I decided to take this opportunity to present oncology and computational oncology and try to establish whether we can, I mean, trigger some interaction with those from PRAIRIE who are not yet involved in computational biology, if not oncology. So those, I mean, familiar with computational biology might find it a bit, I mean, too simple, at least at the beginning. But those who wish to establish links with us, I will try to provide them with elements to understand what kind of questions that we have. Can you hear me well? Yeah. Okay. So. Okay, so my outline will be as follows. I will introduce briefly the Institute on Computational Oncology, talk a bit about cancer, since it's devoted to non-specialists, on computational oncology research, and then present two projects that we completed recently for the first one, and which is still ongoing for the second one, on, I mean, lung cancer treatment. Yeah. So Curie is a foundation, so we have a private status. So we collaborate, of course, with academia and governmental agencies. And we have three main missions: research, care, and teaching. And we cover more or less all types of cancer, not all of them, but I mean, many of them. And one of our particularities is that we are a comprehensive cancer center, which means that we cover both basic research on, in particular, the biology of the cell, the normal cell, but also the pathological cell, and translational research and clinical research with, in particular, clinical trials, many clinical trials ongoing right now. And of course, treatment. We have 3 hospitals and 3 campuses also for research. And a wide range of activities in science, from biology to chemistry to physics and computational oncology, I mean, modeling. We have 5 chairs in AI, 5 chairs from PRAIRIE, of course, that you might know: Thomas Walter, Irene Buva, Chloé Azencott, and Hervé Zambert, and me. So how are we organized for computational oncology today? Well, the first thing, if you want to do computational oncology, is, of course, I mean, to have your data ready for that. And for that, we are lucky enough to have electronic health records, fully electronic for now 24 years, and to have digitized pathology. So pathology is something which will come during my talk again. So it's something very important. It's simply when you treat cancer, one of the first exams that you do is to do a biopsy or a surgery of the tumor and to examine a slice of the tumor under the microscope. You stain the slice and you examine the structure. And what you see is at the cellular level, if not subcellular level, on tissular level as well. And it gives you a lot of information about the severity of the disease, whether it's invasive or not, and it's useful in the first exam to determine that it's really a cancer, so it's a diagnosis, and then to follow the evolution is also an important examination. So it's now fully digitized. It's not the case everywhere in France, in Europe, or in the world. Not all hospitals have that, and it allows us to launch large projects based on an image as well. We have many core facilities for producing data. As you know, computational people, they like data, in particular at different levels, molecular level, but also cellular, tisular, and other levels. And in particular, sequencing, we generate now in a routine basis, we sequence a lot of patients from Curie for the clinical care of these patients, providing us with original and new data. And to ensure, the availability of this data, we have two main bodies. One is the core facility for mathematics, which is about 20 people, plus 50 people embedded in the research in Curie, and the data direction. Mathematics deals mainly with omics data, while the direction is putting in form all the other clinical information that we might use to conduct our research, and of course for the care as well. And we do have this computational oncology department, which altogether is a bit more than 100 persons involved in systems biology, pharmacology, epidemiology, and a lot of machine learning, and both at the level of molecules and images. We also have a local high-performance computing, but we rely on collaboration also for that, in particular with Barcelona Supercomputing Center, which is one of the biggest in Europe, and the national infrastructure, and of course, like Jean-Zé, for example, and collaboration with private companies. And last but not least, we are involved, as I said before, in Paris Santé Campus on PRAIRIE, which is for us, a very welcome and incredible opportunities of collaboration and of recruiting brilliant people. So cancer now. Just a few words about the disease. Well, I guess you all know that it's a disease where cells become abnormal and start to proliferate with no control, and eventually to invade nearby tissue, leading to invasion, and if not metastasis, meaning setting up in remote organs. And it occurs after a mutation of DNA in some cells due, for example, to stress like UVs or chemicals or others. And little by little, it might result in hyperplasia, meaning something which is abnormal but not yet cancerous, like a nevus. And then it can become cancerous. Altogether, it requires at least 5 mutations to become a cancer. So somehow it's a DNA disease. That's my first point. And it is due to mutation. This is not 100% true, but it's quite true in most cases. It's mutations which trigger cancer. But how do they work? Well, what they do is to destabilize the equilibrium of different processes which take place into the cell, like regulating cell division, regulating cell death, regulating differentiation of the cell. And by disrupting them, they push the cell into a tumoral phenotype. And this is controlled by biological networks. And networks are this type of drawing, are represented by this type of drawing here, where you have many proteins or genes which interact together in a complex manner, and end up by regulating processes like proliferation or death. And the networks are in general almost always redundant. You have parallel pathways, and also have a lot of feedback mechanisms to control homeostasis of the cells. So they are very robust, which is a good point, when they're functioning well, and an handicap when you try to disrupt them to kill an abnormal cell. So somehow cancer is also a biological network disease. And then in a normal setting, the immune system, as soon as a cancerous cell will appear, it will be eliminated, killed by specialized, detected and then killed by specialized immune cells. And if it is not the case, it's because it managed to disrupt also the immunity protection by modifying the microenvironment or having a direct impact on immune cells. So somehow it's also a microenvironment of the tumor and immunity disease. And as you can see on the main drawing on the top right, it's a tissue disease. It disrupts completely the organization of a tissue, and that's what possible invasion and metastasis with also tissue disease. So the question is how to bring all of this knowledge together with patient medical information into mathematical modeling for predicting tumor evolution. Okay, so what data do we have when we are talking about cancer, and at least in a biomedical setting? Well, the journey of the patient will go from prevention to detection, diagnosis, prognosis, treatment, and then monitoring of the treatment. And during all of this process, we collect a lot of different types of data. Now, the usual, whole style clinical data, like, the past of the patient, the treatment undergone, some biochemical assays, the medical everyday records, and so on. And this is something which is helpful. Then we have different types of images. One are what you probably know, CT scans, MRI, PET scans, which gives you an image of the whole body, or at least at the organ level, and is useful for detection, of course, but also for diagnosis and prognosis. It brings some information on for monitoring. A third level is at the level of tissular, cellular, and subcellular level. That's the biological slides I mentioned before. That's the essential examination to diagnose cancer and to decide which treatment to adopt afterwards. And finally, another level is the molecular level with all what we call omics data, the GNS or RNA sequencing, but also other levels of molecules, which now are used in routine in cancer care and treatment, the prevention, detection, diagnosis, prognosis, and monitoring also of the disease. So it's really everywhere now. And last but not least, but not fully yet adopted, at least on the research side in our team, are the real-life variables. Everything you can measure using wearable devices like connected watches or smartphone and so on. And if you look at how these have been used according to the different edges of medicine, well, we had several edges. The first one was a symptom-based medicine, which was pretty much descriptive of the symptoms. Molière is one of the most telling example of this type of medicine. The second level was the cause-based medicine, which was developed. The most prominent figure was probably Pasteur, identifying in particular the microbial causes of many different diseases in humans and in other species. And then for like 3 decades, we have a burst of the mechanism-based medicine, where we are able to figure out why some disease is caused, and in particular at the molecular level, where to identify which mutation of genes will cause the disease, and the gene being then perturbed and leading to unfunctional proteins. And we know now to use this information to design small molecules like this red one here, which will interact with this blue protein to repair the malfunction or to destroy the effect of the protein. And now we are entering the data-driven medicine era with artificial intelligence and machine learning. And that's what I will be talking about now, with the idea that we want also to understand and to explain. And the 2 examples I will give are different perspectives on these constraints. And the question that we raise is how to marry machine learning and mechanistic approaches. So mechanistic approaches versus machine learning is something which is not new at all in biology. For long, we had an approach where we generated a lot of profiles of the tumors, information about the tumors, whether it is images or molecular information like sequences, and using machine learning for analysis. It started probably like more than 2.5 decades ago, and it has been very useful and successful to predict some behaviors and to classify at least some simple tasks of classification, not all of them, but some simple tasks were nicely predicted with these approaches. And then, another approach now is to do some mechanistic modeling of the tumors with the claim that we know enough about the internals of the system to build models which explicitly reproduce these mechanisms. And these mechanisms typically are our knowledge of the biological networks I mentioned before, which govern cell death, cell proliferation, cell differentiation, cell adhesion, and all essential processes in tumorigenesis. And this is model-driven. It might be sometimes a bit abstract, but it's useful in particular to explain the result of what we observe and the result of the intervention we can try to bring to the system, like medical intervention, for example. Okay, our approach is to try to, conciliate these two approaches, and as many others, of course, in the community. Okay, so what kind of application do we have in oncology today? Well, maybe the first list of application, I will mention tumor subtyping. The more we are able to characterize tumor, the more we have, the newer technology we use to decipher the molecular aspect of the phenotypic aspect of tumor, the more we identify new subtypes, and the more, medical options that we have, and the more subtypes we need to have to classify patients and give them the best treatment option. So it's very important things, and it's not, it's the unsupervised classification problem. And then we have the supervised classification problems with diagnosis and prognosis and prediction of treatment response, which occupy a lot of our time here in Curie. So there are many other questions in dealing with image analysis, and we have two specialists in Curie with Irene Buvard for MRI and scanners, and Thomas Walter for pathological slides. Thomas has spoken, I guess, to this seminar a few weeks ago. He gave an excellent introduction to these questions. And then there are many other, applications, linked to drug design and repurposing, and more fundamental aspects like deciphering biological pathways or annotating all the molecular information we can get from the large-scale sequencing projects and so on. And still, a few years ago, the most used techniques in terms of methodology in omics were supervector matching and random forest. There have been a study on that a few years ago. And the reason for that is that we didn't have that much data in the field of omics, at least in cancer. So we were limited to techniques which can cope with a limited number of data. But now we have much more data, in particular through new technologies of sequencing, and we're turning for the last 5 years a lot to deep learning. It's really a revolution now in omics, which comes a bit later than in imaging because for obvious reasons, but which is taking place for the last few years. Okay, so I mentioned this data before, and how to make this medical decision from the data. So I will present you an example of project which is based on multimodal information, and multimodality as of today is not as used much in the clinics, at least for developing biomarkers, for really taking the final decisions. In general, the biomarker that we use, to define whether this patient will answer to this drug is based only on one modality. And oncologists will, of course, they have at their disposal different types of different modes of information. Images at this different level I mentioned before, but the integration will be done by the medical doctor in his or her brain with the decision tree schema that he has in mind. So that's not quite elaborated way, but very efficient way still to take decision. Nevertheless, it develops, and for a few cancer indication, we already have multimodal biomarkers developed, which are used in clinical routine now, in particular, for example, for medulloblastoma patients, where we use DNA methylation profiles plus RNA expression profiles to classify in a combined way to classify medulloblastoma patients and determine which type of treatment to give to this patient. So I won't go into detail, but it corresponds to different classes with very different behaviors in terms of treatment response and severity of the disease. So in terms of acceptable side effects, it makes a big difference. Multimodality, so we mentioned, that is different level of modality before with omics data at the molecular level, pathomics radiomics and clinical data. The surprising thing somehow is to understand that they have a lot of overlapping information. For example, if you look at the radiomic data, the CT scan, you can, with quite reasonable AUC value, predict the presence of infiltrating specialized immune cells, CD8 cells. So it's something which to me, the first time I learned that, it was surprising to me, but the AUC can reach 0.75 or 8. It's really something significant. And it's true for many other types of modalities, in particular in radiomic and transcriptomics, for example, but also others like mutations and Wolf-Slide pathological images, or DNA repair impairment and pathological slide. That's the work by Thomas Walter again. And from the pathological slide analysis, you can predict whether some particular pathway of DNA repair is efficient or not. And of course, depending on the response, you can orient the treatment with a particular therapeutic molecule or another. Okay, just very rapidly now, one thing to keep in mind is that all these diverse types of data, all these diverse modality are really heterogeneous in many terms, in terms of dimension, several orders of magnitude, in terms of types, in terms of coverage of the genome when there are, for example, big data. Sometimes you cover very well the genome, for example, with mutation, but not with epigenomics, for example. In terms of distribution, in terms of signal to noise, well, in many terms, they are very, very different, which makes multimodal integration delicate and multimodal complex model construction quite tricky. So now I will give you an example of application of this multimodal type of analysis in a non-small cell lung cancer. It's something which was published last month in Nature Communication, and it's a joint work with Irène Buva, a specialist of radiomics, also a PRAIRIE chair, Nicolas Girard, a lung oncologist, and Thomas Walter, whom I mentioned before, a specialist of digital pathology and other bioimaging, and also a PRAIRIE chair. And I want to stress the fact that a large part of the work on the first authorship is Nicolas Captier, who was a PhD student, funded by PRAIRIE, and we have been lucky enough to recruit an incredibly brilliant PhD student. And it's really thanks to PRAIRIE funding. Without PRAIRIE funding, clearly it will be very difficult to have the reactivity and the flexibility needed to grasp opportunities like very brilliant AI PhD student. It will be quite difficult, if not impossible. So lung cancer, it's not the most common cancer, but it's the most deadly one. And one of the reasons for that is that in more than 2 cases out of 3, it is diagnosed at the metastatic stage, and it's very difficult to cure any metastatic cancer. But recently, the landscape of treatment has changed dramatically with the introduction of new therapeutic molecules, what we call the checkpoint inhibitor-based immunotherapies. So this concept is based on what I mentioned before about the immune system. The immune system should detect a tumoral cell because they are different from normal cells, and that's the job of the immune system to detect what is different from our self and to get rid of it. But to avoid too strong reactions from the immune system, we also have checkpoints. Meaning proteins produced by our body, which limit the reaction of the immune system to avoid autoimmune diseases, to avoid having the immune system killing also our own cells. And when the immune system does not kill anymore the tumoral cells, well, one of the reasons is that we do not manage to unleash it enough. And by inhibiting these checkpoints, which we have this ability to boost the immune response and to be more efficient against killing tumoral cells. Of course, it has to be used with caution because we don't want to trigger an autoimmune disease in replacement of the cancer. There have been some dramatic experiences of that at the beginning of immunotherapies, but now I mean, medical doctors know how to handle that. And it's really a revolution. And by revolution, I mean that now all patients diagnosed with lung cancer at metastatic stage, well, if they express this checkpoint, this protein checkpoint that I mentioned before, they are given this inhibitor of this checkpoint. And if they do not express it high enough, they are given these inhibitors plus some chemotherapy to be safer about having an effect on the tumor. And if you look at the result of these treatments, what is shown on this slide is in PRAIRIE-PSAI, you have the months after the start of the treatment. And in ordinate, you have the percentage of surviving patients. So you see that when you are treated with the checkpoint inhibitor in blue, you survive much longer, at least in percentage. Many more patients will survive longer than if you're not treated with this immunotherapy. But as you can see, it's not full success, and many will not respond. And in fact, almost half of the patients present an objective response, which means half of them do not. And the duration of the response is very variable. Of course, there are some side effects, so it's important to be able to predict who will answer and who will not answer. And also another point is that even those who do not express the checkpoint at a high level, some of them respond less than those that who express this immune checkpoint, but still like 20% of those who are not expected to respond because they do not express the target of the drug, they do respond. So we need biomarkers to predict this answer. And for that, several have been proposed, like the expression of this checkpoint, but we just said it's not that efficient. But others, like how many mutations you might have in the tumor. I mean, the more mutation you have, the more, I mean, it is supposed to trigger the immune system, or I mean, the presence of immune cell infiltrates and so on. They are suboptimal, so we decided to go for trying to build a new biomarker based on multimodal data. So what we did is to, I mean, collect data from a Curie cohort of patients, all with metastatic lung cancer, and all with naive tumors at the time of treatment, meaning they have not undergone any other treatments before, which will modify, of course, the tumors and make, I mean, the analysis much more delicate afterwards. And we gather all the data, the multimodal ones that I mentioned before, at the baseline before the immunotherapy. And of course, we monitor the outcome, overall survival and progression-free survival using scanners, CT scanners. So patients arrive at the institute. They undergo this PET scan imaging to establish the dissemination of the tumor. Some biopsy is taken from the lung in most cases, and stained to examine the aspect of the tumor, and we use this image afterwards for as part of our composite biomarker. And part of the biopsy is used to do transcriptome sequencing, RNA sequencing, to generate a transcriptome profile of the tumor. And we monitor the response of the patient. Does the tumor increase in size or not? That's what we take as monitoring. Okay, and there I want to emphasize a point which linked to the fact that we deal with real clinical data which are collected on the fly, meaning that we are not in the same situation as that you might experience when you work with, I mean, data available in a large-scale international databases or databases like ImageNet. Is really very, very different in the sense that we have many limitations as to data. This slide is a bit outdated, but the main message remains the same. We have collected on this web diagram, I mean, the number of samples that we have for each modality. And as you can see, the number of samples we have for the 4 modalities is limited with respect to the total number of samples we have included in the study. So it's progressing, but we reach better numbers, but we will never be able to have a complete data. If we reach more than 200, I mean, patients with all the data, it will be already a nice achievement. So there is a stronger attrition rate in between, I mean, the number of patients you include and the number of patients for which you have a complete data set. And I mean, first, but not last, we also have a very limited number of samples because it's only a few hundred. And if you pose clinical questions with which are, it's always the case to have this very limited number of samples. We have missing modalities, and we also have for some modalities some missing data. For example, we won't have all clinical data for many reasons. That's real life. So which strategy to use? The first one is which output to predict. And there I want to emphasize the fact that that's really the first question that we have to raise. We don't have enough information to predict, for example, the survival time. We have to define some cutoff. And this cutoff, we have to binarize the data. This binarization should be useful to the medical doctor. And clearly, it's always a discussion with a specialist of the medical question to establish what is the best strategy. So we went for 6 months of progression-free survival and 1 year overall survival, because the medical doctor has to get an information which is useful to him. At the end of the day, he has to decide whether he or she will treat the patient with chemotherapy alone or not. Uncrafted or learned features, so that was also a question, but given the paucity of the data, we went for uncrafted features. And as to the fusion scheme, given that we have a lot of missing modalities, cases of missing modalities, we went for late fusion. Okay, so how does it work? Well, we each of us in this partnership had a specialty. So here in Buva was a specialist of radiomics, and for each PET scan, segmentation was done and extraction of uncrafted radiomic feature, dealing with the whole image and ending with something like a few dozens of features for these for the different modalities that we have. It was the same for pathological images. This one was handled by our specialist, Thomas Walter. And again, it was handcrafted features given the low number of slides that we had. I'm not saying it's the only possible approach, but that's the one we adopted in that project. And finally, for this omics data, we also wanted to reduce the dimensions. So there are different ways of doing that. We wanted to be specific to lung cancer. There are not so many data about lung cancer to learn from external sources of information is not that obvious. At least it was not that obvious 5 years ago when we started the project. Now we start having other possibilities, and probably today we will conduct the project in another way. Basing our strategy also on available external profiles. But what we did is, to select uncrafted features, which are specific oncogenes involved in lung cancer, first things, and also using an extraction of the profiles of the importance of the profiles of different types of immune cells within this matrix of expression. So using signature of each type of immune cell, finding the proportion of each type of immune cells within the transcriptome profiles. Okay, and then we went for simply, given that we have many missing modalities late fusion, given we have a reduced number of patients, simply balanced random forest approach for computing and calibrating the score of prediction. We tested a lot of different things, including more complex schemes, but at the end of the day, this simple scheme was the most efficient, so there was no reason to make it more complex. And this slide is just showing one of the final results of the study, which is on this slide, you have the AUC in different situations as a function of the modality that you use. So on the left, you have the multimodal cases with clinical, radiomic, pathomic, and RNA, and then the bimodal, the 6 bimodal, and then the trimodal, and then the quadrimodal. And what appears is that, the more you add modalities, the better the AUC, even with, appropriate corrections. And another point is that the error bars are still quite large. And another lesson is that if you look at what brings the most information, you can see that RNA is always very, very informative about what can be extracted from these from this data as to prediction of immune response. Okay, so for this part, what we ended, I mean, what we concluded from the study is that multimodality is useful in immunotherapy response prediction. And simple schemes are what we can afford today, given the paucity of data that we have and the particularities of clinical information. And transcriptome is the most informative information. It's still an ongoing project, and now we are looking for external cohorts as well, in addition to adding new samples, external cohorts as well to make sure that what we develop in-house can generalize well and to adapt, I mean, the scheme as a function of what we can learn from other data sets. Okay, and one thing I want to stress again here is that there are many publications in the field of computational oncology on developing this type of biomarkers. And there was this provocative paper by Florian Markovets, one of the prominent figures in computational in Europe, that all models are wrong. Well, that we know, I mean, the sentence by I think it was P.O. Box, and yours are useless, meaning that most of these biomarkers which have been published have not reached the clinics. So I will give you at the end a few, recommendation to make it happen, that it goes to the clinic. Okay. So now I will go for the second project that I wanted to introduce today. Unless somebody wants to ask a question on the first part, or so let's move to the second project. This one is about modeling tumor progression, and it is based on a double methodology. One is agent-based, and the other one is Boolean network modeling. And the specialist in our team is Laurence Calzone and also Vincent Noël. So the first thing is that in what I've shown, I mean, up to now, we have not considered really space as a variable into the game. I mean, and space is really essential to understand cancer for different reasons. First, I mean, cancer is not a tumor is not something homogeneous; it's heterogeneous. There are clones on subclones. The microenvironment is of the utmost importance. We have seen the importance of the immune system in combating the cancer, but also, I mean, the physical organization. Here you have an example of tumors with these blue tumoral cells and some other specialized structural cells which are called fibroblasts. In yellow, they are not elongated. They are not tumoral cells, but they isolate the tumor in some cases from the rest of the environment, and they prevent, for example, immune cells to reach the internal of the tumor and to kill the tumor. So this organization is essential if you want to understand why the tumor is developing and why it will or will not answer to a medical treatment. And this organization is also controlled by signaling networks. And as I said before, these networks have a lot of feedback mechanisms, a lot of redundant solutions to pass a signal. And they're highly non-linear and non-obvious in their functioning. So what we need is multi-omics and multi-scale spatial modeling of these tumors. So how do we do that? We went for a solution based on agent-based modeling, in which each cell is modeled as an agent with its own functioning rules and with some biophysical rules of interactions with the other cell and with the environment. So it can be the diffusion of molecules, it can be the displacement, I mean, the migration of cells, it can be cell division or cell death. All of that is managed in a biophysical terms using, I mean, usual diffusion equation and mechanical equation. This is based on a tool which is developed by one of our collaborators at Indiana University. The name is PhysicCell. But that's the biophysics of the cell. And of course, we also want to introduce all the genetics of the cell, the biochemistry of the cell. And for that, each cell, internal functioning is governed by a network that we are able to model with a tool that we have developed in the lab, MABOS, Markovian Boolean stochastic simulator. And these recapitulate the interaction of the different genes and proteins and the final outcome of the network, which can be in terms of cell death or survival, of proliferation, or adhesion, or migration, or any other important phenotypes, which are then input into the biophysical level of simulation. So by doing so, we are able to model both levels, the biochemical level and the biophysical level, and to have them interact together. To initialize this network, to figure out how they function in the particular cell, what we use, of course, is omics profile, meaning we know how to measure in a tumor the expression level of the cell is something that we can use to initiate this and parameterize this network, both initiation and parameterization. And then if we want to model the effect of a drug, at least a targeted drug, by targeted drug, I mean a drug which will target a specific proteins that we won't have a systemic effect on all of the cell, but we should target a specific protein. Then it's easily modeled as a perturbation of the network that we can simulate with our MABOS simulator. And of course, the gradient and the diffusion of the drug can also be modeled using the biophysical parameters. So it's something that we have used in the past. And here I give a simple toy example where we have different cells, red and blue, red being very adhesive. And if you make the system evolve, you see that they end up at the center of the picture, whereas none of the less adhesive are pushed at the borders of the system. And to figure out whether it works well or not, we had to figure out whether it recapitulates some important mechanisms of cancer progression. So we started with, I will give you an example about the invasion mechanisms. In the context of invasion of a tumor and invading the surrounding tissues, it can do it in several ways. It has to go through a network of fibers, extracellular matrices, and it can be done in several ways that is shown on this slide and at the bottom part. Either cell by cell, it breaches out the extracellular matrix, or in clusters, that's more, drastic way, and that's more serious cancer in that case, or, one by one, but following each other in a trail way. So this is 3 different ways which correspond to 3 different severity of the disease when we talk about cancer migration and cancer invasion. And it's something which, for example, in breast cancer, we can see if you examine pathological slides, you have in situ cases, which is the case on the upper line. You have, invasive cases in the middle line of slides, and you also have the microinvasive, which are in between. So this corresponds to the different types of migration. So what we did is to build a network to recapitulate the interaction with the extracellular matrix and the invasion process, and to model it using this agent-based plus network modeling approach. And by doing so, we were able to recapitulate different types of experimental results that was produced by other teams. This one is by Friedel. He has grown some cancer cells in a medium which is collagen, which mimic the extracellular matrix, and has shown that in some cases it will migrate as a bulk, as a cluster of cells, and in some cases it migrates like individual cells, like single migration. And the modeling that we have performed is able to recapitulate this situation. So this was done in this case, but it was also done in another case with a breast cancer cell line, which was a collaboration with colleagues at Institut Curie, Philippe Chavrier and Salomon, grown again in this type of medium and with different types of gene activation and modification. And what can be done is that if some particular proteins are done, then there is no migration. That's what is shown on the black image at the bottom, which we also recapitulate by our simulation. Whereas if it is on, then it starts migrating and invading the surroundings, which is on the upper part of the slide. And last but not least, we also try to show that it works with another type of activation, another gene, the sarconcoprotein, which is activated through optogenetics, well, through. And when you activate it, that's black images on this slide. At hour 0, there is no proliferation, and then it starts proliferating and invading the surroundings. And when you stop the illumination with the blue light, it reverses the process, which is also what we managed to recapitulate. So now we are continuing, for other aspects of cell progression toward tumorigenesis to conduct this type of analysis. And in order to check whether we can recapitulate these different phenomena which are involved in tumorigenesis and build a simulation of the whole tumor itself. One thing that we did on this data is to simulate systematically all pairwise perturbation of the proteins involved in the molecular networks that we modeled. And we found out that overexpression of one protein, P633, and knockout of one gene, ERK, will disrupt completely the proliferative phenotypes of the tumor. So this is something which now has to go to in vivo testing. And if it works, and if ERK can be used as a drug with no dramatic side effect, it's a therapeutic option, of course. Okay, so how do we go to a higher level of modeling? Well, now we have new technologies to sequence gene expression. Before, what was possible was what we call the bulk gene expression sequencing, meaning sequencing all the cell at once and having one vector of gene expression for the whole tumor, which is, of course, an average view. Then we have this single cell approach. We know how to sequence cell by cell, but only a limited number of cells, not all the cells, and without having kept the precise location of the cell. We just know that we have a distribution of cell gene expression profile, but we don't have the location of each individual cell. But now we have technologies which enables to sequence in situ or to transfer information about the location of the cell, meaning that we can establish almost cell by cell or group of cell per group of cell, a full expression profile while keeping the position of this cell. So we have a spatial full transcriptome view of the gene expression program. So it's new data. It was the technology of the year, like two or 3 years ago, a few years ago, maybe 4. And it's really something we need to be able to convert into biological knowledge and medical practice now. Here you have an example of how it works. I mean, you have this pathological slide. You extract the RNA in all these small spots, and then you have a distribution of expression for each gene. That's for one particular gene on this slide, but you have for each of the 20,000 genes, you will have this type of image on the slide. And well, one thing which is more and more done today is to use tumor, and to do a biopsy and to cultivate, to grow these biopsies to build organoids. Meaning that we try to reproduce a somehow functional small organ, which recapitulates the functioning typically of the lung if you're talking about the lung. And it's something we can do at the lab, and then you can test drug on these organoids, which is derived from the patients, and it allows to test treatment in vitro and to take therapeutic decisions. And the idea behind this spatialomics approach is if you are able to establish the full expression profile, spatial expression profile of your tumor in 2D, then you can do this simulation and figure out in silico which treatments, which combinatorial treatment to do, which you can then apply for either for therapeutic decision or for orienting the test of organoids. You can do it combinatorially here. It's much more difficult to do it combinatorially with organoids because, of course, it's in vitro. Okay, so take a method. This composite modeling is a computational parallel to organoids, and it enables you, with respect to pure machine learning-based approaches, to shed light on mechanisms, and it can be used to orient experiment design and if not therapeutic strategies in the future. For that, we have an ongoing project. It's a project on immunotherapy with Leipzig University. And it requires a lot of computational power, which is also what we do in a EC project. I mean, to convert these tools into something which can be run in parallel and on Mare Nostrum in Barcelona Supercomputing Center, for example, on very highly parallel architecture. So I underline again the role of Laurence Calzone in that work, and Marco Ruscone was the PhD student behind this work. Do I have 2 minutes left, or should I stop here?
I think it's fine if you take 2 minutes more for the take-home messages.
Okay.
Go ahead.
So I will skip this one. Well, just as you have understood, I guess, we are trying to bring to build bridges between mechanistic approaches and learning approaches. And on this slide, I just recapitulate a few ideas that we and others had about what one side could bring to the others. And in particular, mechanistic modeling is quite heavy in terms of computation. So building surrogate models using their results to learn, I mean, a model based on more, less, I mean, CPU-intensive approaches will be quite desirable. It can bring also explanation in interpreting learning models in mechanistic terms. And of course, I mean, once you have a mechanistic model, it's also something you can use to inform your learning. And conversely, in this mechanistic model, we have a lot of parameters. Many of them are unknown, of course, most of them are unknown, and meaning that we have to explore the parameter space, and it's something where learning can be useful. And of course, once we get the simulation results, in general, we don't have one single model. We have a families of models, of mechanistic models, and we need to analyze all the results of these, I mean, families of models. And there again, learning could be useful. If one of you is familiar with one of these questions, I'll be happy to discuss with him. And I just also want to raise one point and propose you one question. Well, what we know is that in biology, in particular, for example, in colon cancer, there is a strong organization, a spatial organization, which is useful for the organ to play its role. I mean, it's not only useful; it's necessary for the organ to play its role. For example, for colon, you have these scripts where this kind of fractal shape, which I mean multiply by 1,000 the surface of interaction between I mean, the internal of the colon and the membrane of the colon. This is very important. It is also a mechanism to renew the colon. The colon is renewed, I mean, the epithelial layer of the colon is renewed entirely every 3 or 4 days. So it has to be very efficient in renewing that. And this organization is also useful for that. So, and it's visible. In tumor, with the aspect of that is that it looks very disorganized, but probably there are some organizational principles behind this disorganization, because it's also subject to a selective process. I mean, the tumor has to thrive, else it will die. It has to, I mean, be sure that to be supplied with nutrients and so on. So there should be some organization which can be explored, for example, with this spatial information I mentioned before, the spatial omics I mentioned before. But as of today, we fall short in finding good ways of exploring. I mean, identifying automatically somehow principle of organization, of spatial organization. So if you have experience in that, I'll be happy to discuss that with you, because of course then it will be an opportunity to develop new therapeutics. Okay. That I have already exposed before, so I will stop here, and I'll be happy to take questions if there are questions.
Thanks a lot, Emmanuel, for the great talk, and you can raise the hand if you have questions. Maybe I can start with one just so that other people prepare for the question for the multi-omics part. So you show that, for example, in the case of lung cancer, depending on the combination of omics that you were using, you could get better or worse performances. What's your advice? What's the best thing to do? Like, especially in situations where we don't know the output. I mean, we don't have a way to measure the output, like cancer subtyping or other type of task. How to choose which omics to consider, and what are the best practices according to you?
Well, if you don't know your output, of course, the general rule, you are in a ill-posed problem. So I guess I only have ill solutions for that. But I mean, very often in the field of oncology, though you try, I mean, indeed, to find automatically subtypes, you have an idea behind that. And typically, the idea will be, I want to have something which will be useful for medical practice. And if you take many of the papers which were published in after, I mean, the TCGI effort, which was sequencing like 50,000 tumors, if you look at the subtyping that they do, what they checked while choosing, I mean, the parameters of their unsupervised classification, is that they like to check whether it makes sense in terms of biology, or if not in terms of medicine, in terms of so they cheat a bit. That's my point.
Okay.
And for example, I mean, I remember a paper on breast cancer where the options between, of course, large possibilities in terms of parameters, and they figure out whether the resulting subtypes were homogeneous in terms of DNA mutations. So okay, so it's not fully supervised. It's not supervised, but still it's
You have some guidelines that can help you.
by some supervision. There is clearly some data leak. If then you want to pretend that it reflects the DNA mutation landscape, you have cheated before.
Okay.
So
I see Anais has a raise hand. Nice.
So thank you very much. I do have a question to know a bit more about what you think about hybrid approaches that would be in between the modeling and yeah. So for instance, I was thinking about the physics-informed deep learning or I don't know, knowledge graph-driven classification and these kind of things.
yeah, so that's the case. It's not really mechanistic driven. You mean it's informed by prior biological knowledge. Okay. Which is something
Yeah,
which...
sometimes also I was thinking at what, for instance, Carl Herrmann is doing when he's doing a neural network, but the neural network is not fully connected layers. It would be connected only by the gene ontology, you know, this kind of thing. So yeah, it's not only prior knowledge, but it's trying to really fuse the two types of approaches.
Yeah. We have tried similar things, but I'm not an expert of that field. But what my impression is that unfortunately, it's less efficient than purely non-informed approaches. If you have a precise question like predicting something, treatment response, often it's less efficient. We have tried to use network-oriented, meaning using biological networks, I mean protein interaction networks, to shape the structure of a neural network to predict the response to treatment, to targeted treatments, with Loïc Verlang, a former medical doctor and PhD student here. In our hands, we did not manage to get better results than purely blind approaches. So I'm not saying that it's impossible, but unfortunately, we have not discovered how to do that yet. At least what I know. You might know better than me. I don't know.
No, I don't know. It's something we are trying to do, but because in the rare diseases we have a problem of number of samples, so maybe it might work better because we have these few samples. Meaning, for me, seeing like you have 100 of samples is like a dream. So.
Well, but that's very little. For many approaches, that's very little. And there are many ways of using this prior knowledge. I mean, and, probably our hope is that using it, for example, in the regularization or in the loss, might maybe be less efficient than a blind approach, but might increase the generalization power of
and interpretability.
Yeah, but that's I mean, whoa, but I don't know if it's we don't have enough experience to say it's true.
Okay, other questions? hesitate to raise the hand. Maybe I can. I have one additional question on your view on organoids, because you mentioned the use of organoids as a possible tool to test therapies in order to give the right treatment to patient, and then possibly the use of modeling to avoid that. What's your view on the use of organoids for these, and to which extent you think they are a good model, let's say, for testing treatments?
You mean the organoids or the
The organoids.
organoids or the simulation?
The organoids.
The organoids themselves. As usual, when you fight cancer, I mean, organoids bring some information, which is not 100%, I mean, precise and correct. So it brings information. It allows to orient the treatment. But the success rate, I guess, suppose it's very dependent on the type of organoid and on the cancer case, as usual. But I don't have, I mean, figures on that. It's really presented by biologists as big hope in orienting patient treatment. Medical doctors, I mean, they are desperately seeking for more information to better treat patients. On any, I mean, piece of information which can increase a bit the reliability of what they propose to the patient is welcome to them. And it's really a step forward, because, I mean, you have to understand that before we used to cell lines, meaning a simple cells, I mean, grown in a medical culture. And but I mean, you take a tumoral cell, you grow it. It is, I mean, you don't have anymore all of its environment, and you pretend that the drug, which is efficient against the drug against the cell in this cell line culture, will also be efficient in the context of the organ in vivo. We know that the microenvironment of the tumor is essential in the development of the tumor and the response of the tumor to treatment. That's this question of space I mentioned before. If you, where is it? This one here. If you extract a blue cell here and treat it with a drug, it might kill the cell easily. But if you take the whole ecosystem, it might not be able to reach the cell itself. So it's really a different problem. So with organoids, we are closer to the in vivo situation. So it's clearly a progress. It's probably not the ultimate solution to have a fully efficient treatment. It's clearly not that either.
Yeah, because you will have a lot of populations of cells that are not represented in the organoid, right? And so that
exactly
gives you a partial view of.
You don't know if your immune cells around are still active or not, are they exhausted or not. You don't know if you can reach a tumor. You don't know. Well, you ignore many things if you only take one tumor cell and grow it and test drug on it. Yeah.
Questions?